Welcome to ASM Live at ICAAC. I'm Michael Schmidt of the Medical University of South Carolina, and this week in microbiology, I'm here at ICAAC, which is the Interscience Conference on Antimicrobial Agents and Chemotherapy, all week here in Denver, Colorado, which is pretty wet and not too sunny like it normally is. But we're delighted to have with us this morning Dr. Kumar from the University of Winnipeg.
University of Manitoba.
Oh, University
Winnipeg.
of Manitoba. I knew I was going to get something wrong. University of Manitoba and Winnipeg, Canada. So thank you very much for joining us. He is going to be sharing with us some of his exciting results that he's going to share with our colleagues here at ICAAC, specifically about oseltamivir and how it can be better used to clear influenza. Dr. Kumar, welcome. I understand you had a harrowing adventure coming in from the airport, which is normally a very pleasant, albeit long ride, but with today's inclement weather, it was a little bit of an adventure.
It was. There's a fair amount of flooding, and they've shut down some of the streets, and it sounds like they may shut down some of the highways later. So.
Oh joy!
Yeah.
Just what we can look forward to.
Absolutely.
So why don't you share with us the bottom line of your exciting research? As we talked a little bit before we went live this morning, you were sharing that many of us have had this discussion back when H1N1 was the pandemic in 2009. I know I personally had the discussion with our state public health department as we were planning for pandemic flu. So.
Well, you know, what happened during that time was we had an early wave of the pandemic, actually in the spring of 2009 in Winnipeg, and we knew we were going to get a larger wave in Canada in the fall, and so we scrambled to put together a randomized trial of standard versus high-dose oseltamivir Tamiflu therapy. Our hope was to get a pretty fair number of patients, and our primary, because we didn't think we could get enough patients to do a clinical endpoint, we thought that a very reasonable endpoint to look at would be viral clearance at day 5, because that's important for two things. One is more rapid viral clearance that you might see, with a higher dose, is often related to survival. But the other reason that it's intrinsically important is because the duration of isolation is related to how long you have virus in your system.
This is especially important for the healthcare working population, the ones that take care of our patients, because we don't want them actively shedding virus as they're taking care of the ill who may not have influenza but have other conditions that will predispose them for a much more severe outcome.
Exactly. So in terms of patients, it has an influence on how long they're infectious to other healthcare workers and other people in the hospital. The other thing the other reason that it's relevant is isolating patients is a tremendous manpower issue, particularly when you're talking about a pandemic situation where manpower is already substantially stressed and may be overloaded. So it's very useful from that point of view.
Yeah, I can well imagine. And the dose that you were using, the standard dose is 75 mgs of oseltamivir twice a day.
That's correct.
And you went up to 225 milligrams twice a day.
Correct.
In your study.
Right, and what we found was, the basics of what we found is, A, that it's very well tolerated, and B, that in fact at day 5, and that was our primary endpoint, viral clearance at day 5, there were many more patients who had cleared virus on the high dose therapy than on the standard dose therapy. The other reason that I think the triple dose therapy is of interest is because with certain other influenza viruses, the ones that are circulating in China, for example, these days, and the ones we're worried about a new pandemic, treatment of those is often done with double or triple dose therapy, and we don't really know how well it's tolerated and what it does.
So the other question that I found important to address with you here is, when did you give the dose? Was it in this magic 2-day window of onset of symptoms that we often hear with prescribing oseltamivir, or did you cheat the system and give it once they seroconverted before they had frank symptoms because they were in your hospital.
Yeah, no, this was, these were patients who presented when they presented. So these were critically ill patients coming in, usually to the emergency room. Occasionally they'd be on the floor for a day or so. But basically, it was whenever they presented. I think the data's pretty good now that, yes, within 2 days of symptoms is ideal, but giving oseltamivir at any point in a critically ill patient is still very useful in terms of outcome. And in fact, our duration of symptoms was certainly not less than 48 hours. It was typically more like 72 hours to 5 days.
And what you really want the oseltamivir to do is to nail that virus inside the cell so you don't have any more release.
That's right.
So that you don't have any subsequent shedding and all of those other issues that you're concerned with.
And disease progression, absolutely. As I said, there's two sides of a coin. One is spreading to other people, and the other is spreading to you and making you sicker than you need to be, basically. So we were very pleased to find that the triple dose therapy seems to eliminate virus significantly more quickly.
So we'll ask you the crass question. Is it good value for money?
Well, for my money, so to speak, I think that it is good value for the money in terms of, I think Tamiflu is relatively inexpensive. The cost of caring for critically ill people, and you have to remember this study was done specifically in critically ill patients in the ICU. That was the criteria for study entry. In those patients, we're spending tremendous amounts of money to support them. And something that clears the virus faster and at least potentially, at least gives you a rationale for, possible better clinical outcomes, I think it's worth the money. I'm not saying that this gives you better clinical outcomes. I am saying, though, the one thing this study does is it gives us a biological rationale for proposing the possibility that higher dose therapy.
And the biological rationale to do an effective blinded clinical trial to understand whether or not single dose label regime is as effective of the triple dose.
Absolutely. And I think a larger study is definitely needed. So this is by no means definitive in the clinical side, but I think it's pretty solid on the viral clearance issue, which, as I said, has to do a lot to do with isolation
and that really is the gold standard that we're really trying to get at with using a neuraminidase inhibitor. Is we're really trying to understand is if the virus is literally shut down, so we don't have to worry about it getting loose.
That's right.
And moving through our healthcare population. The other thing I found intriguing is from the perspective of modeling your health workforce during a pandemic and rationing precious resources like oseltamivir, it may be tremendous value to protect your workforce by managing the population with a regime such as triple therapy, or not triple the dose in the affected population.
That's true. I'm not sure that I'd be as enthusiastic about using triple dose therapy in patients who aren't critically ill because A, we didn't look at that, and B, other people have looked at double dose therapy in non-critically ill people, and they didn't find that there was more rapid clearance. We think that there is actually a difference in terms of response in critically ill patients and non-critically ill patients. A lot of times we kind of almost all of our data on this kind of thing is actually based on ambulatory patients or experimental human infection. And all too often we take that data and we extrapolate it to critically ill patients with the assumption that it applies perfectly well. And our experience with the H1N1 clinical epidemic, some of the observational studies we did, and now this randomized trial really suggests that critically ill patients, for whatever reason, maybe that they have much increased viral load, seem to respond differently than others. And so we need to look at them as kind of a unique patient set.
Well, that's what I found unique about your study is the patient cohort that you were actually looking at. Because I think, as you point out, it's very essential that we begin to look at the therapy that we're applying, because I think they are a distinct population and the drugs may not behave the same way that they behave in the idealized population that they have been effectively developed and evaluated in.
Yeah, I think that's exactly true. The classic example of that is just what you mentioned about, the data showing that really, in the past they've said that if you don't start the oseltamivir within 48 hours, it really has no impact on outcome. And early in the pandemic, people were following that advice as it related to critically ill patients. Now, if you go back and look at the data, and mind you, it's retrospective data, but what you see there is clearly a signal that in critically ill patients, even getting oseltamivir at day 3, 4, or 5, whenever you get it, is good. Whereas if you looked at the ambulatory patient data, you'd say, well, it won't have any impact. So it really suggests that there's clearly a difference. And same thing with the duration of therapy. I think there's good evidence coming in. Our study was 10 days of therapy, and that was because they were critically ill. But again, the old literature for ambulatory patients would say that they only needed 5 days.
Right.
Exactly.
Yeah, I remember having this discussion with my colleagues in South Carolina as we were planning for pandemic flu, and then we were doing the hot wash after the epidemic had subsided, and then we began to evaluate the data on a continuous basis as it came out. And the critically ill population is a unique, we came to the conclusion that the critically ill population was a unique cohort, and that they did indeed warrant oseltamivir beyond this magic window of 48 hours, because judging from the reports that were coming out, it did have a benefit. And again, you like to see outstanding p-values in large sample sizes, but unfortunately in that patient cohort it's very challenging to do that.
No, that's right. I think one of the things about a pandemic, in my experience, is that you learn a lot of things really fast.
Oh yeah, it's like taking information from a fire hose.
That's right, and yeah, you'd love to have a nice randomized controlled trial of anything, but sometimes, you just look at the patient and you look at your experience and you say, I think this is right. Another area where we found something very interesting as an aside is, we were told initially that, if you do nasal swabs on patients with influenza, if it's negative, it's not influenza. And yet we found as time went on that we had patients who were negative by nasal swab, but were positive on tracheal aspirin.
Yeah.
And so that was another thing that we learned literally right in the middle of the pandemic, basically.
And this is going to be interesting because I'm sure you've been following as closely as I have the emergence of H7N9.
Absolutely.
And it's the demon in the closet that the entire developed and developing world are extremely concerned with because of its high death rate.
Yeah.
And the most recent data coming out of China with 130 patients and 44 of them die, I mean, that's an extremely frightening statistic. And so we're really looking for therapies that will not only help our patients but ease the fear that will come should that virus become more readily transmissible. But the other interesting thing about H7N9 is where it's presently infecting, deep. And so there, the nasal swab story
That's right. Yeah.
wouldn't have the same effect. So, looking at all of these things and coupled with your data that you're sharing here at this meeting, I think it's really exciting times because, as you said, the pandemic really offers you an opportunity to really begin to investigate things.
It does. It does. And hopefully there's not another pandemic real soon, but we were real fortunate between the last 40 years, but historically, sometimes it's been as little as 10 years.
Yeah.
So, all bets are off when the next one happens, but it will happen.
Yeah, that's unfortunately true. If you should have any questions out in the Twitterverse, you can reach us at #ICAAC, or if any in the audience have a question, please go to the microphone. We'll be happy to take your questions. We have a question here.
Good morning, Dr. Kumar. I'm John Sheezer. I'm with the Medical Post of Canada.
Hi.
How are you today?
Very good.
Glad you made it through the floods. Yes. I wanted to confirm there were 59 patients in this study,
That's correct.
and 56 were eligible for evaluation,
That's correct.
and 7 out of 9 on the triple therapy were cleared by day 5, but only 1 out of 9 in other arms. Is that correct?
I believe that's correct. Because we didn't have a rapid test available for the study at that time, we operated under the assumption that the majority of patients who would present would have influenza if we caught it during the pandemic wave. Unfortunately, by the time we got set up, we were just a little bit past the pandemic wave. So we ended up enrolling about 60 patients into the study, but only roughly 20 of them were actually confirmed by PCR as having H1N1.
Thank you.
Any other questions from the audience? Dr. Kumar, do you have anything else to share with us?
No, just that, you know, I think the whole H1N1 pandemic experience was quite a driver of research, and I hope, you know, what we're doing will contribute to making the next event milder. I will say that I find it interesting that, you know, sometimes in the lay press you hear that, oh, this was such a mild event. It was not nearly as bad as, you know, they made it out to be. And, you know, I think most of us who work in this area would say that, you know, the reason that it was mild was because we got on top of it quickly and did all the things that we were supposed to do. And it just brings to mind that basically the only time people are going to say, oh, that was really bad is when we do everything wrong.
When
you
it's
know?
when it's really bad.
That's right. You know, and, but if you do everything right, they'll say, oh, that wasn't.
I was involved in our state's planning efforts, and the expression I often used was planning is priceless, and that you really had to make certain that when the vaccine became available, it was to vaccinate as many people as possible, and really begin to follow the simple public health messages of covering your cough and
Yeah.
distancing and staying home when you feel unwell. All the simple low-hanging fruit we were messaging at a very frequent rate, and it did pay handsome dividends. I think the lay public is always expecting, you know, it's sort of like watching NASCAR. They want to see the race car crash, but when it does crash, they're all wringing their hands.
That's
but,
right.
I think we were very fortunate that the public health sector really got on top of H1.
I think so. I think this was much less a mild pandemic than it was a major a public health success, basically. I think it was much more a public health success than mild pandemic.
Well, the thing that it taught me most is how quickly this virus circled the globe.
Yeah.
I mean, it really demonstrated how a high path strain can move without any resistance whatsoever, no matter our best efforts in hand hygiene, no matter our best efforts in distancing. It moved very easily throughout the globe very, very quickly.
It's very interesting when you compare it to the 1918 kind of pandemic when basically you were looking at rail and ship travel. That pandemic played out over a couple of years, basically. Whereas now with the kind of instantaneous jet travel we have to every place in the world, you're right, I mean, it spread incredibly quickly, and it really just goes to show you that just technological changes in terms of travel have a big impact on what would have otherwise been a very atypical, you know, spread. But we hadn't really maybe entirely considered how different a pandemic might look in the age of jet travel as opposed to the age of ship and rail travel.
Well, you know, I think that's one of the most important lessons learned is that if you don't pay it the due attention that it requires, you're going to rue the day, because the virus, you know, takes no prisoners.
Yeah, yeah.
Well, you've been watching ASM Live at ICAAC, and we've been joined here with Dr. Anand Kumar from the University of Manitoba and Winnipeg. And thank you for rushing directly from the airport. We look forward to your paper. It offers tremendous, exciting results. And I'm Michael Schmidt. I'm from the Medical University of South Carolina, and This Week in Microbiology. And we're at ICAAC, the Inter-Science Conference on Antimicrobial Agents and Chemotherapy, here in a very wet Denver, but nevertheless the science is still as exciting as it has been advertised. So thank you very much, and we look forward to visiting with you again.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.